Hi, this is Miss Klemme, and welcome to the podcast on cancer. And over in this corner, I have a picture of the breast cancer ribbon. Cancer is a really emotional topic for a lot of people because most of us know someone who's battled cancer or unfortunately lost their life to cancer. And although it does reach home for a lot of people, today I want to focus on the biology of cancer and how that actually accumulates in someone's body when they're fighting cancer. But first, just to give you a little bit of scope on what we're dealing with here, this is a diagram of different potential risk factors for developing breast cancer. On the outer rim, you can see these are different chemicals and different types of exposures to different things that may cause breast cancer. On the inside are more situational experiences. So when did you have exposure to, let's see, benzene? Was it when you were in early childhood? Was it when you had late menopause, or did you have late menopause? On the inner column, it's what's your diet like? How much alcohol did you consume? Now you can see there's a lot of things here, and there's a lot of things here and up here. Whoa. So, as much as it's really tough to pinpoint things that may have caused breast cancer in this case, you can see there's some areas where there's a little more correlation to, occupational exposure to certain chemicals is pretty high. The air and water quality around you plays a big deal too. So there's some things that you can control, some things that you can't. But bottom line, this is a really complicated storyline to follow. So let's just talk about what happens when you do get cancer. So in a quick definition, what is cancer? Cancer is mitosis gone wild. And that can happen, well, it happens two ways. First of all, your cells just divide too quickly, but usually when they divide too quickly, they also divide improperly. And so that's the bigger portion of this, is that those cells actually are not true functioning cells like our other cells in our body. Now let me refresh your memory on mitosis and the cell cycle. So we're looking at the cell cycle right here. And we can see that we have mitosis, and interphase is that aqua circle. And interphase can be broken up into some subphases, G1, S, and G2. But then we have mitosis, there's the nuclear division, and there's the cytokinesis, the cell division portion. And we're going to look at the checkpoints that are in between each of these phases. So for example, if I can get my cursor, there's a checkpoint here. There's a gene right there that checks the cell, and it makes sure that everything's going okay, that the cell is ready to, in this case, replicate its DNA. There's another checkpoint here that says, did we actually just replicate the DNA properly? And if so, great, let's move on. There's another checkpoint here that says, are we ready to enter mitosis? And those checks and balances are working properly in a normal cell so that it can go through the cell cycle. And here's a bigger picture of the same thing. We have G1, which is an interphase. Let me switch colors here. All right. So here's an interphase, and so here's that first checkpoint that I was talking about. And the gatekeeper at that checkpoint is an oncogene. If you remember, the study of cancer is called oncology, so you can see where onco and oncology are related. Now the job of oncogenes is just to make sure everything's looking good in that cell, and it gives it the green light to divide. That's why we have green light, go. Oncogene gives it that first checkpoint. Now, also at that checkpoint, the oncogene can decide if the cell shouldn't be dividing, in which case it can enter this little side route and maybe even have it commit suicide, which is called apoptosis. If that cell is really messed up, the oncogene can say, you know what, you should not be going on to the next checkpoint. We're going to destroy you. And that's good. That's a good thing. But moving on to the checkpoint, we have a tumor suppressor. And the biggest one that you should know, and we'll talk about in more detail, is called the p53 gene. And these are like the brake pedals. They go through and they check again. Are things going okay in this cell? And if not, if they can fix it, they will. They'll tell some other genes, hey, we need to fix some chromosomes, there's some errors in them, and then the cell can go on and divide. So the tumor suppressor and oncogenes, they work together. They're like a give and take to make sure the cell goes through the cell cycle as it should. So here's again what happens for the p53 gene if it comes across some DNA that's mutated. And remember, a mutation is just a change in DNA. Could be caused by severe exposure to heat, to radiation, to chemicals, you name it. Anyways, it comes across that. Maybe an A is paired with a C rather than a T. And it recognizes that. And so in step 2, it can hire out these repair enzymes to fix them. And if the repair enzymes can fix them, the cell goes on to divide and everything's great. If the enzymes cannot repair the cell, then the cell just goes through and p53 recognizes that. And again, that's that A word, apoptosis. We can't fix it, so you know what, we're just going to get rid of it so it doesn't divide, so it doesn't spread and grow into something that is much more serious of an issue. So that's when everything is working great for the tumor suppressor gene. Now here's the same scenario. Say we have a mutation in step 1, our DNA is not properly replicated. However, unfortunately, now p53 is mutated itself as well, and it can no longer do its job. So when there is an abnormal cell, we can follow it on the bottom here. When it divides, those mutations accumulate, and eventually you'll get a cancerous cell with many mutations in its genes. And that's where things can go wrong. So if there's no p53 working as it should, those cancerous cells have no checkpoint. They just go straight through the gate. So here's the ultimate combo. First of all, we have our brakes cut. p53 gene is no longer stopping cells that have mutations in them. So we don't have any brakes, and then the oncogenes, the gas pedal, those are the gas pedal genes, they're stuck in the down position. So our brakes are cut and our gas pedal is pedal to the metal, and so unfortunately we are speeding close to that finish line, which is cancer. Now those are the 2 key mutations. So I said there's unlimited growth, so we turn on the gas pedal, we turn on the oncogene. Because normally, and I don't know if I mentioned this earlier, the oncogene is something called, before it's mutated, it's called a proto-oncogene. And that's the one that really does regulate the gas pedal. But if that is mutated, then we call it an oncogene. And so then it's on all the time. Moving forward, we ignore our checkpoints, so we ignore the brakes. And if we ignore the brakes, we can't actually then tell cells that are beyond repair to undergo apoptosis. So we can turn off, cancerous cells turn off those genes. They also turn off, or they produce something called, well actually let me show you a picture. So here's our chromosome, and the ends of chromosomes are called telomeres. And every time your cells divide, they lose a little bit of those telomeres. And when they lose the complete telomere, the cell knows, I've been around a long time, I divided a lot, I just better, for the good of everybody here, I just better commit suicide. I better commit apoptosis. And it does that. And again, that is a good thing for your body. But cancerous cells, they actually produce an enzyme called telomerase, which keeps adding on a little bit of telomere. So instead of losing telomere, a little portion of it, it can add it on. So those cancerous cells are immortal. In addition, the cancerous cells promote blood vessel growth around it so that it can get more oxygen and nutrients and grow even faster than regular cells. And the last thing that can happen in cancerous cells is that they don't have any awareness of their surroundings. They don't care. If normal cells have a bunch of other cells around them, they won't divide. However, cancerous cells, they don't have that awareness. So they'll just keep dividing and keep dividing and keep dividing. And that's called, that's what's called, they do not have density dependent inhibition. So they'll just go and go and pile up. And that's where we see the tumors that start. Now, all these mutations that I just listed, the P53, the oncogenes, all of those, they can be caused by a lot of different things, just like that picture I showed you at the beginning of this podcast. They could be caused by UV radiation, cigarette smoke, just your age in general, or even genetics. But the one that the vocab word I do want to bring your attention is carcinogens. These are just chemicals that are known, to cause cancer. So make sure you know that term. All right, so I just talked about tumors and how they don't exhibit density-dependent inhibition. They pile up. Well, there's two types of ways cells can pile up, these abnormal growth. And the first one is the good kind. That's benign tumors. That's just regular cells that have accumulated. Malignant tumors are the cancerous cells, the ones that have mutated in all those different varieties that I showed on 2 slides earlier. So we start with a malignant tumor. And this is in the breast tissue, and if that malignant tumor keeps dividing and growing, it can spread to the lymph tissue. And if it spreads there, it can go all over the entire body. It can start another clump of tumors somewhere else. And the spreading of tumors is called metastasis. And that's what you don't want to hear. You do not want to hear that the cancer has spread to your lymph system, your cancer has spread to your lungs, etc. Some warning signs for cancer. It's pretty open-ended because it really does depend on what type of cancer that you have. So for example, if you have chronic indigestion or change in your bowel movements, perhaps you could have the early signs of colon cancer. Changes in moles, like the color and the shape, could indicate a melanoma, a skin cancer. Bone pain, bone cancer, persistent cough, lung cancer. For detection methods, it's just like those signs and symptoms. It really does matter what type of cancer you have for what type of detection method we're going to use. So for example, if we look at pap smears, that's for cervical cancer, and they'll take a tissue sample of your cervix to see if those cells are cancerous. A generic term for a pap smear is a biopsy, and this is a pretty common practice. If we think that there's a chance the patient has colon cancer, we'll go in, we'll do a colonoscopy, we can take an actual tissue sample and see what that looks like under the microscope. So that's a biopsy. And we can also do some imaging samples as well for breast cancer, mammograms, etc. Cancer treatments. Well, the first one we want to do is get rid of the tumor, get rid of the large growth, and we'll try and get as much as we can with surgery. Then the other two, chemotherapy and radiation, are trying to kill the cancer cells that surgery couldn't remove. And if we're successful, great. But unfortunately, a lot of times those chemicals and that radiation kills healthy cells too. So in addition to some of those types of therapies, you may be on a regimen to boost your immune system so that you have enough healthy cells to keep fighting other infections too. The last type of treatment that many cancer patients have is a palliative treatment. Palliative just really means pain management. So if you're experiencing a lot of pain, you'll be on a strong medication for pain, or maybe you'll have a patch that you'll have to change every few hours that just topically releases pain medication for you. So that's more of a objective approach to cancer, and I hope that you found that helpful. And just remember, cancer is mitosis gone wild. Thanks for listening.